The herpes simplex virus has a large double-stranded DNA genome that carries the genetic information for the synthesis of over 100 polypeptides. A protein capsid surrounds the DNA, and a phospholipid membrane surrounds the capsid. The virus has elaborate glycoprotein spikes that allow it to recognize, bind to, and fuse with a few cell types in the body. The viral envelope becomes incorporated into the host's membrane, and the nucleocapsid uncoats and releases the DNA into the nucleus. In the nucleus, the DNA circularizes, and the host enzymes begin to transcribe the first sets of viral genes, known as early genes. The early mRNA molecules enter the cytoplasm, where they're translated into early proteins. The early proteins, in turn, enter the nucleus, where they're required for viral DNA replication. At this point, the cell begins to transcribe late mRNA molecules. These mRNAs enter the cytoplasm for translation into late proteins. The late proteins include the capsid structural proteins, which enter the nucleus. The late proteins also include envelope proteins, which are produced in the endoplasmic reticulum, ER, and become incorporated in the nuclear membrane. The viral DNA continues to replicate. One model postulates that the viral DNA replicates by a rolling circle mechanism. During this latter stage of replication, end-to-end copies or concatemers of the viral DNA form. These copies are cut into individual genomes as they're packaged into capsids. At the nuclear membrane, the capsid and DNA bud off and become surrounded by membrane material with embedded membrane proteins. The virus passes through the endoplasmic reticulum and buds off this membrane before migrating to the plasma membrane. The virus fuses with the plasma membrane and is released from the cell.